So it's an honor to present Dr. John Sellers for today's student alumni. And John and I arrived here in Kentucky about the same time. We were starting a PhD program with Dr. Collins, and I'm taking a postdoc position with him about the same time. So we've kind of been here this whole time together, and we get to see each other at meetings. Found out after we met each other that we also shared an office at Oklahoma State, although we were kind of separated by a few years. And so both of us were in graduate school at Oklahoma State. And that's where John got his undergraduate degree in agronomy and also his master's degree before he came here with Dr. Aaron Lindsay, the agronomy department. While he was here, he got his PhD and was co-mentored by Dr. Hunt and Dr. Collins. And I got his dissertation title here. So read it: Potavirus NIA protease-derived resistance. Evaluation of resistance and media interaction with the potavirus polymerase. For those that don't know Dr. Hunt, you know the word polymerase written in there. After he left here, he went to North Carolina, where he did a postdoc for a couple of years. And in 1999, took a position as a research molecular biologist at the ARS unit in Manhattan, Kansas, the hardware wheat genetics unit, where he's been ever since. He's also an adjunct faculty at the Department of Plant Pathology at Kansas State. Also the adjunct professor at University of Nottingham as part of the advisory board for the wheat research genetics. And he was recently also kind of detailed as an acting research leader for a unit in North Dakota, part of North Dakota, the cereal crop research unit, where he was the advisor, but the research leader, but where he was in charge of the unit. That also has another member that was was also a PhD program in Dr. Zhu's lab, Sheng Yang. So again, there's the Kentucky connection. We want to welcome Dr. Sellers here to present what our investor invested in, how we invested in him for his research career.
All right, can you guys hear me? All right, great. Yeah, what an honor it is to be here. And I have to tell you, the reason why I'm standing here today is really goes back to Dr. Egley. And it was I was at the agronomy meetings trying to figure out what I was going to do for my PhD, and universities had their table set up. Hadn't even had Kentucky on my mind. And Dr. Egley was there, and he took my information, brought it back to Dr. Collins. And 30 years ago, I got a call from Glenn and started down my path. So again, thank you, Dr. Egley, for that chance that you gave me and gave it to Dr. Collins and pass that along. So I'm glad the luggage didn't get lost that day. So on the way back, I thought it would be interesting. I asked Art what he'd like for me to talk about, and I said I could talk about my research, current, or do you want me to talk about life history and what this is all about? And he said all of the above. So I thought, you guys made an investment. This department made an investment in me. The university made an investment in me. And so I thought I'd give you a little bit, if you think about it from a business perspective, what that money you invested in, what you got out of it. So let's see. Oh, there we go.
Did you get it? Sometimes you just have to touch the John.
Okay.
You hold that.
Okay.
You just have to do this. Now try it.
There we go. Okay.
You got it for
Yep.
Go for it.
All right, so if you get to thinking about what all I've been a part of during my life, my grandmother was born right at the turn of the century, of the 20th century. And one time I sat there and asked her, I was like, you know, grandmother, you've seen a lot. You've seen really electricity getting put out to a lot of rural areas. You've seen the advent of the telephone. You've seen television. You've seen space travel, flight. She saw a lot. And so in my life, when I was in high school, I started working on the Apple II on BASIC. And so had the floppy drives and the green screen and my first computer I worked on, I had a Commodore 64. That was my graduation gift. And that typed all my papers in college and up until my master's. And then now we know what kind of computing that we have available to us. Also mobile phones. When we were here in Kentucky, we had a bag phone. It was one of the first ones that got a great deal from Cellular One. And for those of you around that time, it was quite, you had to have the curly little antenna off your back window, you know, if you wanted to be part of the social norm, you know, everybody wanted to, you could plug these things, you could either get them solid in your car or you could plug these into. We've seen satellite navigation. So I'm a pilot. And when I got my pilot's license while an undergrad at Oklahoma State, and it was all by magnetic compass and by big maps. Now it's all on my iPad or my phone, and I can literally fly anywhere and come through the clouds and be within about 6 inches of where we want to know. And anybody that's done any field work knows that we can get precision now down to mere inches when planting out in the field. Recombinant DNA, we've seen what we can do with DNA and how you can move DNA from one organism to the next. You can clone it. One of the things Art Hunt always threatened on me was to make me isolate my own EcoR1 for bacteria. But luckily we didn't have to do it. We still, though, every once in a while make our own Taq polymerase. So one of the big things that really helped us all out is DNA sequencing. And I still like showing the autorads from my work here at Kentucky to my students and show them and tell them how long it would take a week to get one of these if you were lucky that it worked. And then also plant transformation. And so right before, while I was at a master's, the gene gun had been invented. And my master's project was to try to look at developing embryogenic suspensions of wheat as a target for the gene gun. Cool thing about the gene gun was you used a 22 cartridge at that time, so it was literally the gun. And when I got here, Collins had just retrofitted his to using helium. So a lot has happened. So what Randy didn't mention is I'm a farm boy from Northwest Oklahoma, wheat and cattle farm. I started driving a Ford 8N tractor raking hay at 8 years old. And so I've been working on the farm. My family came from, or my great-great-grandfather came from around Dayton, Ohio during the land runs of 1893. If you've ever seen the movie of Tom Cruise, Far and Away, it talks about the land runs. That's how my family got to Oklahoma. And so he homesteaded 320 acres, of which I own today. And my daughter has driven a tractor on it. So it's 6 generations right now. And so I have a really unique perspective. I still actively farm with my family. Most of what we have now is rented out, but we still have about 500 acres that we still grow in wheat. And usually I spend my vacation time in June running a combine. So like Randy said, I got my BS at Oklahoma State and crop science option. And I was the first student to graduate with the biotech option. And I worked as an undergraduate with Dr. Eric Lindsay. He was fresh out of his PhD from Kansas State. And the whole world of tissue culture was coming on. And the idea of biotechnology was happening. And we were working on herbicide tolerance by putting herbicide in the media and getting plants that would grow in herbicide. So, and then the gene gun had come out. And so I was plating immature embryos from wheat onto plates, and Aaron was taking them up to Pioneer, and Pioneer was blasting them. And this work led to Dr. Troy Weeks, who was a PhD student in our lab. He was the first one to publish using the gene gun and wheat transformation and from stuff. So I've been a part of that. So during my master's degree, like I said, I worked in wheat suspension cultures, and we got a little article in the High Plains Journal published because I was one of the first ones to get wheat to regenerate from these suspension cultures. And so at the time, we didn't know we could use immature embryos. We were hoping to use suspension cultures. And so this was taken about this time, about in March, 30 years ago. So this is another example of what 30 years of science can do to you if you look at me now. Luckily, I have a mask on, so you don't see that much. But anyway, I worked with David Porter. We used two-dimensional gels in identifying proteins that were unique to embryogenic callus. I remember at the crop science meeting, somebody coming up to me and said, "You'll never do 2D gels again." And then later, about 10 years ago, people started making robots to pick the spots out of 2D gels to identify the proteins that were there. So never believe what somebody told you at a poster session. But along about February, like I say, Dr. Egley had gotten my top of my information handed off to Glenn Collins. He called me up, said, "We'd like to have you out." An interview. I came out here. Like I said, Kentucky honestly was just not on my radar. I was trying to get in with Wayne Parrott at Georgia, but I was 0.05 away from the grade requirement. I couldn't get in, and but I was looking for more skills and working in the transformation and trying to apply biotechnology because I really wanted to improve crops. I mean, I knew there was a new way and new opportunities. So that's what I was really hoping. So I defended my thesis about 30 years ago this next week, and then moved to Kentucky right here in August. Like I said, Glenn and Art were my co-PIs. Did most of my work in arts lab. The committee was Saeed Gabriel, Mark Nielsen, Mike Salvucci, and Joe Chappelle. And it was also a transition time for tobacco research. This was also the time when the Clinton administration decided to shut down all wheat research or tobacco research. So Mike got transferred to Arizona to work on stress. So Joe Chappelle took over his. So this picture I got actually at Glenn's memorial. It's probably one of the latest pictures of Glenn's group that we have, because Santanu's in there, and it's you never know your connections. I was actually driving a tractor, listening to NPR one night, and they were talking to a scientist with a company in India about the use of biotechnology and Santanu. And I was like, I know that guy. I thought his voice was really familiar. So you just never know when and Phil Ray here was actually on Jeopardy. So one time, so before he left here. But anyway, I got 3 papers out of here, and one of them I'm really proud of is the transformation from the main work of my work, but it was Art's first crop science paper. So I got him that accomplishment. I also had with Yiling Hong, we worked on a protein-protein interaction. And then the gene gun focusing device was something where we took pipette tip and rigged it into the gene gun so that it would focus the particles into a very precise area. I still have that piece of plastic and I still use it today. So it didn't stay in Kentucky. I took it with me. So my oldest daughter was born here, and I defended in September, and then on the 19th, on the 24th, we packed up and moved to North Carolina, and then did my postdoc, and then moved to Kansas in December of 1998. So that's kind of my University of Kentucky here. I was here for 4 years. It's just a wonderful time. So what have I been doing since? One of the things that we've been working on is I work in the hard winter wheat genetics research unit. We are integrated into the department, much like Randy's unit is integrated into your department. There's 2 of us in plant pathology, 3 in agronomy, and 1 in entomology, but we all work on weed improvement. So we're directly tied with Alan Fritz and the wheat breeding group, the Wheat Genetics Resource Center. Our breeders, we do a lot of pre-breeding in what we do. I'm more of the genomics and molecular biology person. I established our first sequencing facility for wheat. But one of the big things that I'm really proud of, and is working with Dave Van Sanford, is the idea of being able to set up regional molecular marker centers for wheat. And so Gina Brown Gadara, who is a UK alum, and we wrote a white paper talking about the idea with Dave's help, and then Dave took it to Capitol Hill, and we got it funded. And so now we have these 4 regional marker labs that give service to the plant breeders to help them in what they do. I've also been part of some 3 big cloning projects with the sequencing lab. The Q gene is the wheat domestication gene. It prevents the shape of the spike. It prevents it from shattering. It holds on to the seeds actually, and it was one of the most important genes for the domestication of wheat. LR21 was the first wheat disease resistance gene to be cloned, and I did that with Vikram Gill. He did all the mapping, and I did all the sequencing for it. TSN1 was with Justin Ferris. It's unique in that the pathogen actually uses the resistance gene. The pathogen is a necrotroph. And so it uses the plant's resistance pathways to cause the cells to die, whereas the plant would usually do it to isolate the pathogen. So it's pretty cool how the pathogen's kind of outsmarting it. So that was a lot of fun. Now I'm working on 2 other genes. Like I said, we worked on the first wheat backs we sequenced in my lab. I've done the red flower beetle. So anytime you've opened up your flower box and you've seen a little red beetle in there, that's the tribolium. It's a pest and stored grain. We sequenced, got early start on that genome project. We did the Hessian fly. Done 3 rusts now of wheat rusts, and now I'm starting to work on some of the wheat wild relatives, and then some other wheat work. Virology, I've continued. I went for a pause there, but wheat has 2 podoviruses that are very popular. So my podovirus work that I did here has come into work. Now talk a little bit today about our field population work. Working on pathogen host interactions. I did a lot of early work in fusarium. Students, when I first, it was an embarrassing moment, my first meeting where I was kind of introducing myself to the wheat world, one of the fusarium workshops. All through the talk, I had fusarium spelled wrong. I had 2 S's in it. And it wasn't until a friend of mine, I sat down, he goes, "You realize fusarium's wrong in your whole talk." So just an embarrassing moment. So looking at leaf rust and viruses, and then I won't talk much about it today, but and looking at susceptibility. How do pathogens get into it? I talked here about 3 years ago, and I talked a lot about that work, but I'm not going to talk much about it today. Also doing some alternative approaches, and this is kind of building from some of the work that I did here and applying it to wheat, looking at pathogen-derived resistance, looking at RNAi, and then we're also starting to employ some CRISPR applications for virus resistance. And then this is work that I'm doing with the University of Nottingham about integrating some genome of wild species of wheat as a source of either new disease resistance genes or new environmental tolerance genes. But anyway, so I want to talk, since I did some virus work here, and this is really kind of something new that's popped up here in the last year or 2 in my lab, is I want to talk about wheat viruses. And so particularly the big one that we deal with is the wheat streak mosaic complex. It's a complex of multiple viruses. They all have the same phenotypes if you looked at the viruses individually. It's wheat streak mosaic, triticum mosaic virus, high plains wheat mosaic virus, and these are responsible for 5% loss total in the US and worldwide. But on a local level, it can wipe out a whole field. And I'll show you a picture of just one of our research plots and how devastating it can be. Another one is Barley Elegor, and this is transmitted by an aphid. There's one isolate that does aphid, one is the green bug. And so this one, we don't see, this usually happens in the fall, but you don't see the symptoms until later. And it looks like the K-State folks like it because it makes the leaf purple. So the K-State Wildcats are purple. First question I got when I moved to Kentucky, they said, or moved to Kansas, they said, "Are you a Wildcat fan?" And I was like, "Not the purple." So, but anyway, this is one of the devastating viruses that we deal with, and a little bit of the problem that we have is that, like for triticum mosaic virus, we just don't have any easy detection kits like the ELISA kits. We don't have one for triticum. They're commercially available. You can make your own, but that causes a lot of variation. So with the diagnostic, there's several issues. Is the diagnostic kits are only available for the viruses that we know of, and they're also very specific on virus titer. So sometimes you got to have a big virus titer. Anybody that's done a COVID test, okay, and shoved the wand up your nose. You've got to have a certain amount of virus titer for that kit to work. And it's the same idea that we're dealing with in the wheat viruses. You can do PCRs to look at SNPs, but then you have to go through, you have to know the region of the genome, you have to know some sequence information. You've got to do isolations. If it's an RNA virus, you got to move it over, you got to change it to DNA. So there's a lot of steps that go through it. Then if you don't know much about the virus, you have to sequence it. And the old way of doing it is clone it up in small pieces. You use Sanger sequencing and piece it all back together. Well, that's very tedious and long. And so that's one of the problems. Also, with the new technology like Alumni that sequences short pieces, if you're wanting to understand the big virus, those short pieces, when you put them together, it may not have a good representation of what the actual virus is causing, or the version of the virus is the way I should say it, whether that's the right one, or whether it's kind of made a piecemeal of all the virus pieces. It's okay, and it gives you a general idea, but for the specificity of that particular virus genome, it's not very good at it. And like I say, it's all of this is based on the assumption that only the tested virus is present. Okay, that means, let's say, if we didn't have, if you having symptoms and you went in and you only had the test for COVID, they would test for COVID and it would say negative, right? It might be flu, or flu A or flu B. Unless you have a test for that, you don't know. So in wheat and what we were doing, we only have tests for certain viruses. So that's one of the big assumptions. Another issue is that most of the time in wheat breeding, we look for resistance genes as our best way to combat infections. Wheat virus resistance genes are few and difficult to find, and they come from wild species. And so we only have 3, and one of them has already gone down for wheat streak. The Barley Elegor is a chromosome integration, but it has some agronomic traits that aren't good that come along with it. So they're hard to find. And like I said, in 2017, we found a new isolate of WSMV that overcame this. And then the big question that I asked the plant breeders, yeah, you're selecting for wheat streak mosaic virus resistance, but are you sure that's the only virus in the field? So we don't know. Some of the other work is, can we use pathogen-derived resistance and use parts of the virus against it? And a little bit, can we look at the susceptibility, or what genes are involved that the virus needs when it infects the plant, that commandeers from the plant, can we use those against the virus? So this is what the two potiviruses look like. This is Wheat Streak Mosaic virus. This is Triticum Mosaic virus. We didn't know about this until around 2006. But as you can see, you would, if you looked at the plant, the symptoms are the same, and you couldn't tell them apart. If you look at the structure, they look like a potivirus. They have their flexus rods. This is Wheat Streak that we got from the University of Lincoln, Nebraska at Lincoln. This is work that I did with Triticum Mosaic and out of my work. These are called these spaghetti shots because it does look like spaghetti, but it has the typical structure of the genome, one large open reading frame that has 10 amino acids that break apart by self-encoded proteases. So something I was I knew a lot about. So, and they have co-protein and a poly-A tail. So of course, doing what we, this is not new idea about using the co-protein, except that we have used RNAi of the co-protein instead of just an open reading frame. And so this is with Triticum Mosaic virus. So we created some transgenic lines. So this is Bob White, which is the variety. There's only two varieties of wheat that we can really use for transformation. This is Bob White. It's a spring wheat out of Mexico. This is what inoculated Bob White looks like with Triticum Mosaic virus. It really wipes it out. So this is a T5, that means 5 generations after transformation line with the co-protein. You can see that it looks really well. The question was, okay, this is a spring wheat. It doesn't work in Kansas. Let's cross it into a Kansas variety. So we cross it into Oberle. So this is the F1. This is the back cross F1. And this is what Oberle looks like by itself with the virus. So the co-protein works and it works well. Unfortunately, trading partners don't want the GMO. You guys, there is co-protein squash, there's papaya. And so in certain systems, it's been accepted, but in wheat, nobody wants it. So we have Roundup Ready wheat on the shelf. I have all these virus resistant lines on the shelf, and it's just, we can't go anywhere with them. So we try to look at other ways. The virus itself has a VPG here on the end. This VPG simulates a cap structure that like of the RNAs in the plant, and it recruits this whole complex. And so this complex has several initiation factors for G, F2, F3, 1A, and all of these bind here and start translating the virus. And so the virus needs to be translated. It has to uncode, it translates, and for every co-protein that it needs to bind up the viral RNA, it's got to make one of these. So this VPG binds to a 4E or an isoform of this 4E, depending on the system, Arabidopsis, but it's very common with potiviruses, both in dicots and monocots. So what we decided to do was to make silence lines using RNAi to silence the isoform of E2 and also for 4G. And these are T5. The reason why we go to T5 is that wheat is very susceptible to methylation of transgenes. And so we carry them out to T5 and test them at every generation, because usually by T2 generation, a lot of the lines, the gene that we've transformed in there is shut off. So what this represents is that this is the gene, the 4-iso-E2. These are the events, transgenic events. This is the number of plants that we tested. This is the number of plants that when we tested had the gene of interest. These are the number of these plants that were resistant to wheat streak, number of them that were resistant to Triticum Mosaic. And then when we did co-infection, this is the number. So it looks like that by silencing iso4E, we also did 4G, and we got very similar results where we saw good resistance And data that I'm not showing today is we also tested this with Barley Elegor, and we saw a reduction in Barley Elegor and also a soil-borne mosaic. So it's more of a broad spectrum. So this was these two genes. Harold Trick, who I met while I was at Kentucky. He was doing a soybean project with Glenn Collins as a postdoc. He and I are now working together at Kansas State. He's my transformation guy. But this is what one of those plants looks like. This is the Silas line. This is the infected line. But we did get 2 patents out of this technology. So this year, or this last summer, I hope it's, I tried to blow it up as big as we could. This is in Lane County in Kansas. This is our Wheat Street Mosaic Virus Nursery. And this is how serious of an infection we got. So LCS Chrome is a line that was derived by Marla Hall, who is a graduate of UK. She now works for Lima Green as their wheat breeder for the Midwest. And it's very susceptible. This is one of Alan Fritz's experimental lines. It's a TAM 114. It does have a resistance gene in it. Joe has WSM2 in it. Everest doesn't have a resistance gene in it. And WB Groundfield is our susceptible check. But as you can see, you can go out there and say, okay, yeah, this one has virus symptoms in it. This one does. This is triticale, which is a wheat rye hybrid. This one does not have any symptoms on it. So the virus symptoms, we can say, okay, yeah, it's this, but it could also be abiotic stress, soil chemical toxicity, nutrient deficiency, heat, drought, and it can mimic these kind of symptoms. It's difficult to base them, and then in most cases, to verify this, we base it on PCR ELISA. Again, assuming that the tests that we have are the only viruses that are in there. So when we saw Clara CL, we got a sample in from Eric DeWolf in 2015. Clara CL has WSM2. It was just released. It's a clear field wheat, which means it's resistant to herbicide through a mutation, not through transgenic. And came in, and this is my daughter Madison, who was born here. She was a unpaid volunteer that summer in my lab, but she did all the PCR, she did all the cloning through the classical method, and she assembled the genome, and she got her name actually on 2 papers from that work that summer. So I'm really proud of her. But the way she did it was that we knew it was Wheat Street, so we were able to design PCR primers and PCR small fragments across the genome. And this worked, and then as you can see, they're overlapping. So then we went into a computer program, or with the PCR. These are the fragments that we were able to amplify. She cloned them. She picked 5 colonies per clone. We sequenced them. She put it in the computer. And this whole process is what she did all summer just to get this answer. Okay. So what she found was there were 2 virus species or strains in there that were 98 and 90 or 98.6 and 98.5 to WSM. This type for this is Sydney 81, which was collected in 1981. And so this one, the big one here, was that there was a 3 nucleotide deletion that was in this one. So we pretty much decided that yes, it worked. We were able to find the virus, and we think it was we have 2 different isolates in there that we think might be overcome WSM2. Well, along about this time, I was trying a new technology in my lab using the Oxford nanopore system. And like I said, with Madison's project, if things worked well, it would take 6 weeks to find the presence of the 2 genotypes. For the Oxford nanopore, from the sample to sequence, from the time we got the sample in the lab, isolating the RNA, make cDNA, do the reaction, put it on there. It took a day and a half, and within 2 hours, we knew, but there was enough runs that go through there. We knew that we had wheat streak. And by 24 hours later, we had a 40x coverage to assemble the genome, and we knew that we had several isolates in there. The Minion device is the size of our harmonica. Okay, the Sanger sequencer, and I'll show a picture of it here in just a little bit, that I first started out with was the size of a refrigerator and cost $350,000 in 1999. So this is the size of it. It fits in my pocket. I could take it with me. People are taking it in their tents in the Amazon to sequence all the organisms in the soil in certain parts of the looking at the biome, the various biomes that they have. They're using it in guts, looking at the gut biome in animals. So the device is $1,000, flow cells $900, sequencing runs $100. You have a small PCR machine, 2,500. You can use your laptop that you have, and the new fungal cell, which is much smaller, is only $90. So it's a really for detection, it's very cheap. So the next year, in 2017, we got more samples in from lines. The lines that have CL here are the Clearfield wheat that I was talking about. These lines all have WSM2. These were collected from, this is the Clara CL that Madison worked with. We went back and resequenced it with using this technology. So we had Hamilton County, Greeley, Wichita, and Wallace. This is the total number of reads from a single run, the total amount of base pairs that were isolated, the average length of the reads, since this was cDNA. And from what we found out was that not only did we find WSMB in that sample that Madison sequenced, but also TrimD was in there. This is the X coverage that we had for each one of those viruses. For the Greeley County, we found that it had wheat streak, TrimD, and BYDV. And it also, even as low as 150,000 reads, we could detect that wheat streak, TrimD, and BYDV was out there. So this started changing the whole thought process about what the population is. This last one is Carl 92, which is that we infected with this MHK isolate that we have in the lab, just to use as a kind of as a control. So because we had so much sequence, we were able to assemble a whole genome. These are long reads. The reads that I use this technology in my leaf rust fungal genome sequencing. Our N50s are 30 KB reads. Okay, the virus itself is 10 KB. So now in one run, we can sequence the whole virus of one strand. And so this allows us to, we don't have to worry about the fact that they're getting pieced together. We can do the whole thing. And so this, so what we found was that in the replicase, we found a nucleotide change here that translated into an amino acid change that we think is the causal mutation. It's the one that's most common. So my colleague at University of Nebraska-Lincoln is now taking this and using his expression vectors to verify that this is the actual causal. So Dave, this is the picture I wanted to show you. So this is the rest of that Lane County. And so these are the lines, how devastating the virus was that last summer. These are triticale check plots. For some reason, triticale is happy. These particular lines here have the WSM1 gene. This one has the WSM3. 1 and 3 come from Robertsonian translocations from Thinopyrum elongatum. But they also bring along with them a lot of extra DNA that has some agronomic properties that aren't great. So what makes these so special, and really what's going on in these plants? So we actually, and I hope you can read that from the back, I teach a seminar course, and this is an example of what not to do. But with the Oxford Nanopore system, you can barcode. So we're able to run 12 samples in one run to help with cost. So what this represents is the table of in that replicated trial, the individual wheat lines that are there. This is the barcode number that was applied to it. This is the total reads that was sequenced from that sample and that one run. So the number of total viruses that was in that barcode, the percent of the total run here, this is the percent that this particular sample represented. The number of reads that actually map to a reference set of serial viruses. So this is the number of the percent of this total of this. So 3.81 was actually virus sequence. And then this is the number of reads for each particular virus. So this is wheat streak mosaic, triticum mosaic, wheat high plains mosaic virus. This is I'm having a brain fart right now. Foxtail mosaic, and this is brome mosaic. This foxtail mosaic, we have found out, this is one of those where we get a poly A signal in there, and we're getting a bunch of false reads that are aligning. As long as it's along the poly A section, it gets a hit, but once you get outside, it doesn't make any sense. But the brome mosaic is real, and this actually nobody had detected this in Kansas before this work. So nobody had even thought about brome mosaic being in Kansas. But what this shows is that triticale, even though it looked as good as it did, it still had high levels of wheat streak mosaic, triticum mosaic. It had some high plains virus and brome mosaic virus in it. So it's still happy, but it still had virus in it. So WB Granfield, like I said, is a susceptible check. It was hammered. It still had a level almost as high as the triticale. You saw what it looked like, but the encouraging ones were the wheat streak 3 and the wheat streak 1. You saw those plots; they look great. This particular line also has a wheat curl mite resistance gene in it, and actually you see that here the wheat streak mosaic virus is reduced, almost no triticum mosaic virus, and then the reciprocal, though, is in Guardian. Guardian does have wheat streak, but it also has a wheat curl mite resistance gene in it. And here we see very little wheat streak. We also see high levels of triticum mosaic. So what this is saying is the wheat streak 2 might be working, but this other virus is going on here. And plus, we don't know what the CMC4 is. We've gone in now and we've got replications of this trial to try to get some sort of statistical meaning of it, but this is really some unpublished early work. So that's kind of my virus work. What else I work on is leaf rust. This is the leaf rust life cycle. It's an obligate biotrope, and most of it has two hosts, wheat, and then a sexual cycle takes place in meadow rue. Meadow rue is not present in the U.S., and so most of what we see is we believe is clonal. It makes Uridina spores. It moves on to the next host, makes more Uridina spores. And then at the end of the season, as the plant dries out, sometimes it makes Telia spores. If the alternate host is present, it will go through an asexual stage, and then the asiospore will go back to wheat. So the way we characterize the leaf rust is one of the most common rusts in the world. And the way we type this and understand what isolates we have is based on a set of isolines. These are wheat lines that differ, have been back crossed so that they only differ by one leaf rust resistance gene. So this is Thatcher LR1, Thatcher LR9, Thatcher LR3KA. So it's a really nice tool to help us understand what isolate we have. So if an extinction agent brings us an isolate, we purify it, we spray it on this, and then based on whether it has a high infection type or a low infection type, it's an assigned a letter based over here. For this example, it's high on LR1, it's low on LR2A and 2C, and it's high on LR3. So we go over here and look, and we see high, low, low, high. So the first letter is M. So all the isolates are typed this way, and we're now out to 5 to be able to look like it. Most of the resistance genes are hypersensitive like, so they're flex. This is what the infection, this is particular LR17. And so, but like most resistance genes and leaf rust, they only last about 5 years before they get overcome by mutation. So without a sexual cycle, how is that happening? That's the big question in my lab. So it's all based on effectors. Most of what we know with the HR type genes, there's an effector. The effector, the pathogen puts in an virulence protein that has a resistance gene or some other, or resistance protein or some other that recognizes that effector, and you get a defense signaling. Sometimes there's control up here that it affects some sort of control mechanism. It has some RNA binding or it modifies the protein, but somehow that effector does something to trigger this resistance. And so in lines like this, or in isolates that have the M, only the effector for LR1 has changed, but it has not changed yet for 2A and 2C. But if you put out 2A and 2C out into the field, then within about 5 years, you also see this. So what kind of mutation has changed? And so that's really this part of my lab. So kind of explain that. So I'd like to take a step back and talk about what sequencing was like. When I first started in the lab, we would take a leaf, we would isolate the DNA, we'd have to cut it up into small pieces and clone it. It was based on blue-white selection. You picked the white colonies, you isolated the DNA from it. We had a nice genetics Q-bot that did that. It would pick about 30,000 colonies a day. It cost $250,000, and within 4 years of buying it, it was obsolete. And a guy bought it for $1 and made a router out of it through K-State surplus. But anyway, you would pick these colonies. Each colony would be picked into a single well and a plate. We'd have another robot that cost $80,000 that would isolate the DNA from these. Then you would take a 96 well plate, do a PCR reaction on it, clean out that reaction, put it on. This is the ABI 3700 that I got when I first got there. It was $350,000. It would do 96 samples every 2 hours. You get 70,000 letters per run. Every run was $1,500 in 1999. And by the end of the day, you get maybe 420,000 letters per day at a cost of $10,000 per day. So the Rhabdomyosis, rice, corn projects were all done by this system. Those were each $50 million projects. The human genome was done this way. It was a $500 million project. Okay, I got a grant for $1 million in 2009 to do leaf rust, and that was when we were moving away from Sanger sequencing to the new second and third generation sequencing. And now I can sequence the leaf rust genome in about $15,000. But anyway, when we got the grant with the Broad Institute, we sequenced it. The software wasn't available. It's a dikaryotic fungus, meaning it has 2 nuclei. Each nuclei has its own genome. But we were only able to assemble a haploid genome. It had almost 15,000 scaffolds. And on those scaffolds were made of almost 25,000 contigs, very chopped up. But we also did gramineous. Gramineous had been done. It was 88 for the haploid. Ours was this size, and then stripe rust was 120,000 for megabases. So even though these rusts are very similar in the way they look, their genome sizes on the haploid were very different. So this is also a slide that I tell my students not to do, but because it got so cheap during that time, we were able to sequence 130 different isolates of leaf rust. We compared it to that haploid genome, and we were able to distinguish different collections and lineages from those. These are Ethiopian Durhams. These are European isolates from Durham. This is Speltoides. We think this is probably one of the early progenitors of rust on cereals. This is Agelop cylindrica, goat grass. This is the isolates that we get off of goat grass, is a different lineage. This is probably a sexual lineage from Europe, but this is the oldest lineage from North America. The race 1 that we sequenced is down here, is our reference. But this is what can happen when you release one resistance gene. This lineage showed up in 1997. This is called the Jagger shift. And then a variety by the name of Jagger was released by Kansas State, had LR37, and this whole new lineage popped up when we released that gene. So anyway, we just published this work in G3. But the question that we wanted to ask is, without a sexual cycle going on, how are they overcoming these disease resistance genes? Where's the variation come from? And talking with Mark a little bit ago, maybe changing some thinking on this. But the thinking always is that it's the effectors that are getting selected. Well, we asked the question, are the effectors or the secreted proteins, is the variation any more specific there than over the whole genome, or is it the whole genome that you see this variation? And according to our test, the answer is no, is that the variation's happening equally across the genome. So that's kind of one of the new things that we came out. So the question, or the issue is we need a good genome. And so with the new technology of sequencing, we like to say we have 2 nuclei. We need to be able to take DNA reads, have them long enough that we can piece them together. The longer the read that you can have, the more accurate the assembly. We needed to have a way to separate DNA or the reads into a nucleus A and a nucleus B. How can we do that? And really, even though I did that work and I got it published using a haploid genome, I'm not real comfortable with it because of the quality of the genome that we had. And so now, and working with Goose Bachran and Sean Forby and Steve Hedlund in Canada, we've used the Oxford nanopore system to sequence, and also a high-C technology that uses the way the chromosomes interact in the nucleus to get long spans across the genome. I won't quite go into it today, but we were able to get in the first assembly, we were able to get 86x coverage using nanopore and PacBio, and we were able to get it down to with a haploid genome of 42 contegs and 146. That was 2 years ago. Software has changed because all other people are dealing with the same thing. So using these long reads, our average read length was close to 25,000. We had some Illumina reads, 150 base pairs, and this is exactly the same isolate, spore maps, DNA as we did in the version 2 that we published with the Broad Institute, and again, all using this new technology. So our new assembly is now we can assemble into nucleus 1 and nucleus 2. The first iteration, we can get it down to 95 contigs from 15,000. And the largest contig is 9.4. We think that's the largest chromosome size. We can go telomere to telomere now with this. And then the genome size of each nucleus is about 125 megabases, which is about our Radalopsis equivalent. Now we can start to go in and actually look to say, okay, if this particular isolate, the M isolate, can't overcome LR2A, 2C, but T can we go in now and look at the genome and say, is that effector right here, is it present in nucleus 1 or is it missing in nucleus 2? What is the change? That's why we really needed a good genome. And so we believe right now that we're about a month or so away of having a chromosome to chromosome or chromosome-based telomere to telomere complete genome of leaf rust. So we're pretty excited about it. Now, then we can go in and start looking to see if we can use GWAS to do associations to try to find effectors for the particular genes, LR1, and keep this just within a specific lineage. But then, so now that we have a genome, we can do that. Another thing is to use some of our genetic tools. We have a nuclear reactor at K-State, and it's cheaper for them to keep it running than to shut it down, according to the Nuclear Regulatory Commission. So I took some spores, the postdoc did, and shoved them down the reactor to do fast neutron to try to knock out these effectors. So this is pustules, or this is one of those mutated races that's sprayed on the LR2C isoline, and we'd isolate a pustule like this, and on LR26, we'd isolate and grow those up. And then we do Illumina sequencing. Then we would test them on the whole isogenic set. It's hard to see there, but this is stature, this is a susceptible parent. And this particular isolate is high on LR2A, LR2C, and it's also high on LR2B, and these are all alleles of one another for the resistance gene. So we think it's probably the same effector. So then when we go into it, we can start looking to see, like this is for LR11. We have two isolates where we just see the virulence shifts go from a zero fleck, which is low infection, to a high infection type. So we have LR26, LR2A, 2C, and we also have some for these. So now that we have a genome, we can actually go in, but we need good variant colors. So, and this is where I'm at right now. So this project, I actually showed this 3 years ago, this same slide. So I stopped it. And once I figured out that the gene reference genome that we had was not good for this. And so all this is sitting there waiting on our genome to get assembled and finished. Another thing that we're dealing with is looking at susceptibility, looking at not just think about resistance genes, but what is the pathogen using from the plant for resistance or for infection, and can we change those proteins that the pathogen uses as more of a broad-based resistance like LR34 and LR67. Those are one's ATPase transporter, the other one's a sucrose transporter, not an NBS LRR resistance gene. So if we can, so I've done a whole screen, I've isolated 25 lines that have, so this is the wild type, this is the F1 between these two, and this is the mutant. So there's no spores here. And if we look at the way the fungus grows on it, this is mutant number 531. This is a time course where we've looked at infection. This is wild type stature. This is the spore. This is the approsorium. This is where it goes into the cell. This is the hypotubes. These are the brain fart. I'll think of it here in a minute. So you can see as it grows through, this is what it looks like by day 4. And then, but in this particular line, it actually induces a resistance-like reaction. So this is the spore. By day 1, it looks fairly normal, but by day 2, you start to see cells die off. The fungus is actually trying to escape. But by day 3 and day 4, you just don't see anything. So we're pretty excited about this. And Mary Gutierrez took 2 of these and crossed them. And so this was Action Bottoms in Kansas in 2019. This is Castroville, which is where our big rust nursery is. And this is the mutant line, or the line that has the mutation in it. It's in a 3-way cross. This is another mutant. This is one of them that I'm most excited about. This is a lesion mimic type resistance. This one is, you see just the reduction in the number of pustules and pustule size. And so in Kansas and a little different background. It's almost clean compared to the other one. So we're kind of excited about this. So what now is we're finishing up the genome. We're identifying SNPs through RNA-seq work. We're looking at susceptibility, weak mutants. And we know that they work in the field, so it would be nice to be able to have some RNAi type transgenics to test these. So to wrap it up, this is my lab right now. Carla Redelia is my new technician. She has a master's in virology. She did her work on looking at field populations of wheat streak. So it's nice not to have to train somebody. They can just take off and go. Joe is my graduate student working on the susceptibility mutants. Craig, Myron, and Vanessa are old postdocs and graduate students that worked on the effectors. My current collaborators are Jim Colmer. He is the leaf rust guru at the Serial Disease Lab in St. Paul, Minnesota. He has the world's collection of leaf rust, so that's where a lot of that sequencing came from. Goose and Sean, I mentioned, assembled, is working on the assembly. Sean is a really good bioinformaticist. Christina Cuomo is our collaborator at Broad. Les Zabo is one. He is the one that has been working with UG99 strains coming out of Africa that we don't have much resistance for. This is kind of the scary business. He just actually retired. David Cook works on Fusarium or Verticillium and also wheat blast with barbivalent. And then Fahey was a postdoc with Edward Ockenhoff. Harold Trick is my transformation guy. And Katie is our bioinformaticist in our unit. I want to say thanks. Sorry. Thanks for taking the chance on. And giving me the opportunity to learn from my failures, learn for giving me a chance to go out and explore. Thanks Art and Glenn for really making me think critically and being long time interested friends. And this is, feel pretty honored because two of your other awardees are in the same picture. Don Mei Shu and Quinn Lee, we were all in Art's lab here, so at the same time. So this is a big honor, and I thank you for it. So I'll take any questions.
Thank you, John. We have time for questions. Does anybody have a question or two? Mark?
Yes.
So you said that you used fast neutrons to develop your mutant. You probably got deletions and not SNPs.
Yeah, that's what I'm hoping.
So you probably can get away from the issues of alignment-based SNP calling by just doing chemochem.
I'd like to be able to think that way, but there was a paper where people did this in rice, and they still found the individual SNPs changes. They found the big deletions, but
Well, you'll want to do a window type counting.
yeah,
approach.
we're looking at 2 kb windows right now and looking at.
So I can explain.
Oh yeah, that's
you
great.
slide your chemo
Yeah,
interrogation across a window, and then you see definite fingerprints of deletion.
yeah. And assuming that every one of those mutants came up individually and are not related to each other, then trying to figure out how to get those overlaps and define it to make sure that we get the right ones. Yeah, but yeah, I like talking about it.
We got one more question from the crowd.
Yeah, Dave.
So John, with wheat streak, I mean, do you see any so-called field resistance like we do with Barley Elegor, where you can't really attribute it to any particular genic sources? You have some genotypes that seem to tolerate pretty well, much as the triticale seems to.
At this point, I don't know of any. I mean, I think Alan would be the one to answer that question. But we have done, so in talking about my EMS mutants that I use for susceptibility for leaf rust, we screened 2,500 for leaf rust. There's still 1,000 of them left that I haven't screened. We screened about 4 of them so far for wheat streak mosaic. And we've actually seen in those where you don't see any kind of symptoms. And that's what we're screening for. So there again, are we really screening for the right thing? And so some of them, you know, I've gone through several rounds of selection and they keep showing in the growth chamber. Usually there's no virus there. We take them out to the greenhouse and the virus shows up. So, you know, is it these genes are temperature sensitive. Is it what's going on that causes it? So that's a good question about, and goes into more of the integration stuff, that as we screen those, whether we need to be looking for that. You know, if I find a plant that doesn't have any Barley Elegor symptoms on it, is that from the integration or is it because of something else? So, but that's a good question.
Well, John, thank you very much for that. And so I have a little plaque to present to you to commemorate that you are our distinguished alumni award winner for this year. As you said, and for those of you who may not know, this award program started when I arrived here. I was actually on the committee with Dennis DeCroning and probably someone I'm forgetting that started this award. And so this is our 15th. One of these, and you're very accurate. You've got several in the photo with you there.
Well, I have to say, Charles Rice was also an award winner.
From
And,
State.
I play softball with him.
Right.
So
And you guys can hang these on the.
that's right. Yeah. So
So congratulations.
thank you very much.